The next talk is from Christoph Spahn. He's a postdoc in the group of Ricardo Enriquez in the Gulbenkian Institute of Sciences in Portugal. He will talk about deep learning for image analysis applied to microbiology. And the title of his talk is Democratized Deep Learning for Bioimage Analysis in Microbiology. Thank you, Christoph, for being here and sharing your work.
Yeah, many thanks for the kind introduction, Maria. Can you all hear me? Okay. Yeah, also big thanks to the organizers for hosting this great meeting and for giving me the chance to talk here. I'm actually a postdoc in the group of Professor Mike Heilemann, but I'm currently doing my EMBO short-term fellowship in the group of Ricardo Henriquez. And I want to show you what we've been working on in the last month, and that is how we can use open source deep learning approaches to analyze microbiology microscopy data. So I'm using super resolution microscopy to study bacterial cells at the nanoscale. And in particular, I'm interested in how the chromosome is organized in bacteria. So for example, how it's segregated and replicated, or how chromosomal structure is perturbed by antibiotic treatments. And by that, I want to explore how the chromosome is organized, but also how the antibiotics work. So while we're getting really good resolution with our techniques, what we're missing is the temporal information. And that's also very interesting, right? To know how the chromosome moves and when specific rearrangements occur. And the reason why we cannot do this is that we use really high laser powers and long acquisition times. So in order to be able to correlate the dynamic information to the high spatial information that we have to somehow circumvent these limitations. And in the recent years, deep learning has really boosted microscopy or analysis of microscopy data, and also contributed to circumvent the mentioned issues. So when you have a closer look, it becomes apparent that the main target for deep learning is higher order organisms or mammalian samples. But there's also a lot of potential for analysis of microbial microscopy data. And that's when I joined the group of Professor Ricardo Henriquez in order to explore how we can use deep learning technology for microbiological data. So I want to present you 4 use cases where I tried this and applied deep learning to data I recorded. And I want to start with a data set where we want to denoise live cell data of chromosomally labeled bacterial cells. But here's first a short introduction in how deep learning differs from classical algorithms. So usually you have in this case, noisy input image of labeled chromosome. Typically, you then apply post-processing algorithms and combine different filters in order to then get an output image that has hopefully an increased quality. So deep learning now takes a different route. So we have a large training set that is split into 2 parts. One part is the low quality image data set, and the other part corresponding high quality image data set. And you feed that into your neural network shown here in the middle. So this has a quite complex architecture consisting out of different layers, and these layers consist out of tens to hundreds of neurons that are connected. And this complexity also gives the network a high flexibility in order to learn the mathematical operations that are required to transform the input image into the desired output image. So once you train your model, you can then use unseen low quality data, insert it into the model, and then predict the corresponding high signal to noise ratio data. So I have to say I'm an experimentalist and not a computer science. So if you want to know more exactly how neural networks work, I warmly recommend to visit the workshop of Romain Lane on Thursday, which will be held on the Zero Cost Deep Learning for Microscopy platform. Okay, but of course it's not so easy. For deep learning, you need expensive hardware, large data sets, long training times. And for people like me, it's almost impossible to implement such a network by yourself. So the community has put a lot of effort in democratizing deep learning, meaning creating environments that allow people with non-computer scientific background to also use deep learning efficiently for your research. And I've been mainly using the zero-cost deep learning for microscopy platform in combination with Google Collaboratory. So back to our question. We now have here the highly resolved bacterial cell, and we want to know how the chromosomes dynamically behave in that cell. So we construct a strain which has a fusion protein that labels the chromosome. So if we then go here for confocal imaging with a high quality imaging setting, we will have quick photobleaching, right? And that's a bit disappointing because we want to follow the process and the dynamics of the chromosome for longer times. So what you can do is you can lower the exposure, but then you lose signal to noise ratio and resolution. And here comes in the deep learning approach, where we can use the model we just trained on fixed cell data and insert our time-lapse series here in order to restore the signal and be able to follow the chromosome dynamics at much higher speed and for longer times. So of course, you have to be sure that you can trust the output of the neural network. And that's also a general criticism that often appears when talking about deep learning or artificial intelligence. But there are tools how to check the performance. And there you can, for example, compare the ground truth data you acquired in a test data set with a prediction of the neural network, and then create error maps to see how good the network performs. And here the errors are actually quite low and the structural similarity is relatively high. So we're quite confident that this is doing a good job here. So what do we gain? So with this, we gain signal to noise ratio and resolution that is comparable to the high quality settings. And we are also able to increase the temporal resolution and decrease the photobleaching quite a lot. And additionally, on top of that, using less laser is of course less phototoxic for your cells, meaning that your results that you obtain are probably, that you can trust the results you obtain with this approach. So but we can go even further. So what if we could acquire fluorescence data or pseudofluorescence data without illuminating our sample with strong laser light? And there has been seminal work by the group of Johnson in the Allen Institute, where they trained neural networks to predict pseudofluorescence images from live cell brightfield image stacks. So of course, bacterial cells have much less features than eukaryotic cells, but still it would be quite nice, for example, to see where the cell membrane is in that case, and especially if that would be on a subpixel accuracy. So we also trained the network here and use brightfield data and corresponding super resolution paint data, where we have a highly resolved image of the cell cylinder here. And by training that model and using it on brightfield data, we can then predict where the membrane of these cells is, not just in untreated cells, but also in cells that were treated with different antibiotics, showing that the network was able to generalize quite well. If you compare that then to the ground truth paint data, you see that there's a strong agreement between those two structures. So the nice thing here is that you can train the model in the cloud using the democratized platforms, and then download the model to infer your brightfield images on your local machine using, for example, the image analysis platform Fiji. And of course, you can also apply this to your live cell images then in order to obtain a highly resolved image of your membrane structures. So what would be really cool then is to create a model zoo for different species of bacteria or microbes in general on different setups to provide a possibility for people that don't have, for example, access to this high resolution technology to also create or predict the membranes of the specimen they are looking to. Of course, with a caution, because you also have to check every time whether the data is artificial or can be trusted. Okay, so what else can you do? One task that is also very important in microbiology is segmentation of your data, meaning to identify where the cells are and create regions of interest. So usually microbiologists use phase contrast here. And there are quite a lot of packages out there where you can segment your data quite nicely. However, there are also cases where this is a bit more complicated. For example, this data set from our collaborators, from Séamus from the Newcastle University and his master student Mia Conduit, where they labeled FTZ, giving you a quite heterogeneous image. So these are Bacillus subtilis cells where the constriction ring is labeled in the center and FTC, but also diffuses in the cytosol. And they wanted to get segmentation masks out of that. And here we failed using the networks I presented previously. So we went to a different kind of network, so-called image-to-image translation neural networks, which are able to generate artificial data, for example, to convert daylight images into night images or sketches into realistic photos. But if you have quite defined training data and a lot of that, you can then also use it to generate images that can be used for downstream segmentation. And by that, for example, also segment this kind of data or also conventional brightfield data that is not the quality of phase contrast images. So in the last example, and this is the most exciting for us, I want to introduce how you can use object recognition. And everybody knows that, right? We know it from face recognition from cameras or also from self-driving cars. And the neural network is here trying to classify parts of the image and to draw bounding boxes to indicate that there's, for example, a car or a pedestrian or a stop sign or whatsoever. So it has been recently shown that this can also be transferred to microscopy by the Eggeling lab, where they used these detection networks and augmented reality to identify cell nuclei and also to automatically control their microscope. So we then try to apply these kind of networks to two different questions. So on the one side, we used antibiotic-treated cells to train the network. So we had a lot of images in which the cells were treated with different antibiotics. We trained the network and then created synthetic images consisting of bacteria treated with different antibiotics. And using the network, we were then able to quite accurately classify the cells in this image. And this has, of course, a huge potential for drug screening applications. And here you also see the link to my research. So I want to use this in the future to also investigate mechanisms of action of antibiotics and drugs. Another thing, and this is now the last example, is you can also use it to classify cell cycle stages of your bacteria. For example, you can train the network to discriminate between rod-shaped cells, dividing cells, or microcolonies. And by this, you can, for example, also tell the microscope when it should capture data. For example, if you're interested in dividing cells, it should search for spots where you have a lot of different cells that undergo cell division, or if it should avoid places with many microcolonies. You could just tell this to the microscope, and then it can autonomically acquire data in large amount that is then very well suited to perform your analysis and research. And I think this is the future where microscopy is going to, automating processes based on assumptions that you tell the microscope beforehand. And I'm really curious where this will end. And with this, I want to summarize my talk. I hope I could show you that there's huge potential for deep learning in very different kinds of applications that can be used by people that also have not that detailed experience in deep learning or a strong computer vision background. And of course, those networks can also be combined, not just as shown for the individual tasks, but maybe also to predict outlines from brightfield images that are classified at the same time and then go to high temporal resolution imaging of, for example, chromosome dynamics. Okay, with this, I want to close and thank the Heilmann and Henriques group, especially the people involved in the zero-cost deep learning for microscopy project, like Romain Lane and Lucas von Chamier, also the collaborators, Séamus Holden and Guillaume Jacquemin, the funding, especially the EMBO for the short-term fellowship support, and you for, of course, the entire open source deep learning community, and you for the attention. Thank you.
Okay, thank you, Christoph, for your talk. Impressive, and the applications and potential that it has. So we have quite a lot of questions here, so I will try to summarize. So, well, we have the first questions from Pedro Matos. His great talk regarding the cell membrane labeling. How accurately can you determine the progression of septal closing? And also the second question that is related to that, have you tried to correlate membrane labeling with protein localization?
Yeah, so those are both great questions. So regarding the first question, it depends which stage of cell division the cells are in. So the beginning you cannot track that nicely because the invaginations are quite small, but once you the constriction closes, you can also see that in the membrane image. So I have to admit that we did not have too much training data to also cover this cell cycle stage precisely, but I guess it can be, yeah, if you inject enough training data, you can also get a quite nice prediction there. And the second question was about protein localization. So we tried that for MREB, and that actually gave quite promising results, although we still have to work on that. Though I guess it's possible to get protein localization if the protein is somehow correlated to the membrane structure. But for example, for soluble protein, that might be a bit more difficult.
Mm-hmm. Okay, thanks. And another question from Genaro Saavedra. He's also congratulating you. And what about the false alarms in blind inspection? He means with no ground truth available. Can you say something about the false positive, negative rate, and so on?
Okay, so I've been mainly using supervised neural networks where you have the low quality and high quality data, and there's also been always a test data set where you know the ground truth. There are approaches where you don't need the ground truth, so which are self-supervised, and we also tested some of them, and especially for the denoising, using noise to void, that was quite accurate. It wasn't so the quality was close to that one given by the supervised neural network. For the antibiotic treatments, for example, that strongly depends on the drug that you're using. So for drugs that give a quite nice change in nucleotide morphology, for example, we get accuracies up to 80–90% for each cell, meaning if you have an image with 10 or 20 cells, you can really nicely predict which antibiotic has been used or which mode of action is behind that antibiotic. If you have two compounds that interfere with, for example, cell wall synthesis, and you get roundish cells, the network can, of course, not so easily discriminate between those, but still it can get the mechanism of action. So meaning there you might have 40–50% true positives, but still you get the right mechanism of action there.
So thank you, and thank you so much for your interesting talk.
Yeah, thank you very much, and also looking forward to more questions in the speaker session.
Yes, sure.